Okay, a final speaker on the area of genetics is Dr. Sam Clarke. So Sam comes to us from Armidale, where he's born and bred on a superfine wool operation up at just near Gyra. He's had a PhD now for 3 years. That's about right. Yep. And he also does some work with lecturing for university in the genetics subjects, but he also helps present the Sheep CRC Genetic Service Provider Training, which we hold down in Melbourne each year. So Sam's talks today is on the roles of breeds in a genomic world, looking at a lot of the association analysis, that some of the work that they and AGBO have done. So Sam Clarke, everyone.
Thanks very much, Luke. So hopefully I'm not too loud with the feedback that we've been getting a little bit with some other speakers. So I'm here to talk to you today about, do breeds matter in a genomic world? First thing I kind of ask myself is, what is a breed? Did it matter before the genomics world came along? Really, breeds can be grouped in what they've been bred for, whether it's been animals bred for wool, animals bred for milk or maternal qualities, or is it animals bred for meat or terminal qualities? And then, of course, we have a few animals that have been bred for not having any wool at all, and bred for meat quality as well, and different types of things. So breeds are just groups of animals that have been bred for a certain purpose. So when we come into a genomics world, we start to think, well, where does the information come from? And we've seen this slide a lot. We have a reference population, we have our selection candidates, and we want to try and predict their breeding value. So we can either genotype them and get our breeding values that way, but this reference population always works on the premise that performance recording is happening. It may not be happening by you as breeders, but someone else on a research station is recording some sort of measurement. And so we could also look at this picture before genomics came along, and this reference population was every single one of you that recorded an animal. And so the only link that linked our selection candidates with that reference population was the pedigree that you recorded. And so it's exactly the same thing. It's just that we may have to record fewer animals for our genomics tests to work. And so we can use that for predicting breeding values, but we can also use it for asking different questions about what are breeds, what are they important, do they all group together, or are breeds actually a really diverse range of animals? So we can do some nifty statistical models. I won't go into them because they're a bit bland. And we can tease out the animals that group together. And so we see at the top of the graph, we have our merinos that group together quite tightly. They're all bred for wool. And we can see they've all come from the same place pretty much. We have our maternals that group together, that are bred for our milk or maternal qualities. Then we have our few terminals that group together. We can also see that in our terminals and our maternals, there's a big spread of actually the types of animals that are in those breeds. So we have down this bottom corner our Pole Dorset animals. In the middle we have some Suffolk, some black-faced Suffolk. And then we have white Suffolks that kind of spread between the two. Some white Suffolks are more Pole Dorset than Suffolk. Some white Suffolks are a bit of everything, because we see that they have some maternal breeding as well as some terminal breeding. And so breeds really, at the end of the day, don't really matter. It's your breeding objective that matters. All these animals are still high performance animals. It doesn't matter if they're a Pole Dorset or a white Suffolk. One interesting thing that happens is that the Texels, which are considered to be quite a terminal animal, are actually grouping with the maternals. And within a lot of the maternal breeds, a lot of Texel has been incorporated into them, because Texels are actually quite good mothers. But when it comes to predicting breeding values from genotypic information from certain breeds, you can see that there's no links between merinos and terminals, and very few links between maternals and terminals. So you can't really use this information to predict a bunch of terminal animals. So when that comes to genomic prediction, we have to have reference populations that are specific to these certain groups so that you can still make accurate predictions. And I know that Sam said that we may get some sequencing and this may help us make the link between the two, but it may not either. That's still a question that is unanswered. So it's it's a research question. We don't know what will happen. But at this stage, we can only predict in those breed groups. So really, breeds are just a term for a lot of animals that have been put together on a common breeding objective. And they're just a bunch of genes that have been selected for. So whether it's for wool, as in merinos, whether it's terminals, which are the meat breeds, and whether it's genes selected for maternal qualities. And they're our main subsets of breeds within the sheep genetics database. So if we have a look at the process of genotyping animals for genomic prediction, our reference population for terminals would be made up of terminal animals only and be used to predict our terminal animal selection candidates only. But then within a breed, there can also be quite a bit of variation. And so how do we actually deal with that? So within merino, is there a lot of difference between, say, a fine wool merino or a broad wool merino, as this picture would kind of lead us to believe? This is what it used to look like. There used to be very, very clear definitions between strains of merino. But of late, especially the last 15 years, there's become a lot more crossing of the strains, and a lot of genes have been transferred between those strains. And so we can see there's less of a clear distinction between each different strain. And that's important when it comes to genomic prediction, because you can be able to use all of this information to predict our merino animals. And so that leads me to think, well, does relationship matter? Does how closely we're related to an animal in the reference population help me as a selection candidate be selected or have a more accurate breeding value? So we can work out the relationship between selection candidates and the reference population with our genotype tests. And we can use some of this information to try and help us try and predict how accurate our breeding values will be. And so at this current stage, we've always talked about a single accuracy for a genomic prediction. And that's what we've been talking about because it's quite simple. But in essence, there's actually more than one accuracy that you could get for a group of animals. And that depends on the relationship between the animals that you've tested and your selection candidates. So if we have a little bit of an example here about how relationships estimated with genomics are different to relationships estimated with pedigree. And so we should be all relatively happy with the concept that our genes come from our mother and father, and it's half mum and half dad. But then if we're compared to our full siblings, in a pedigree system, we assume that we're all about half related, or we share about half the genes. So our two selection candidates are full siblings, and in pedigree we think they're about half. So we use half the amount of information that comes from each other. But in essence, they could actually vary from being 0.4 of their genes being the same to 0.6 of their genes being the same. And using that information within an evaluation system could be quite important, because some full sibs are more related to full sibs than others. And so if you can utilise that information, you get a more accurate prediction of breeding value. So if our full sibs were actually, so we can see here, by pedigree, they'd get all exactly the same number, but with genomics, we start to spread those relationships out. So we can actually start to differentiate between full sibs. Which one's a better full sib than another full sib? And so when that comes to predicting a breeding value in a whole population of selection candidates, we have a range of accuracies that could occur. So what we've been talking about are these baseline accuracies that come from, that everyone within the population can get. But there are some animals that you may actually predict a lot more accurately if they have quite close relatives within the reference population. So if you even just have a cousin in the reference population, you could predict your breeding value much more accurately. This is for everyone, this is for ones that are a bit more related. And it may be one way of actually dealing with smaller breeds as well, is utilising some of these larger relationships to actually help you predict a genomic breeding value. But one problem with using these close relationships is that they break down quite quickly over time. And so the baseline accuracy is what you can rely on to be sustained over many generations. And these larger relationship accuracies that are driven by larger relationships tend to break down as you become less and less related. So this is just a graph to show, similar to what Sam had before, that you need lots of animals to get certain accuracies. And this is just some based on theory, so it's some made up numbers that someone took a bit of a guess knowing what the population would be. But it turns out that we're actually doing a bit better than they thought we would. And so if we record more animals, then you get a little bit more accuracy. So after saying that we can use genomic relationships to estimate breeding values, that's exactly how single step works. So single step can combine pedigree relationships from the past with those estimated using genomic technologies to give us a more accurate estimate using all of the information that's been collected over the last 25 years. So that all of the information that everyone here has collected doesn't go to waste. You still use all of these phenotypes that have been used on sires and grandsires within that single step analysis. And so it gives us more information gives us better results, better predictions of breeding value. So what does this mean? Do breeds really matter? Well, when it comes to prediction, breed groups kind of matter because we can't predict a crossbreed at this stage. But we can't use merinos to predict terminals, we can't use terminals to predict maternals. And one major thing is measurement is still important. Just a matter of figuring out who does the measurement, whether it's you as farmers and breeders still recording some traits, or whether it's Sam organising MLA to fund the reference population of resource animals. It just depends. And whether there's a mix between the two could be quite good as well. So measurement is still important. Okay, that's about it. Thank you very much.
Just going back to that was
Which one?
that one there. Yeah, I think. I'm glad they don't have this at the moment for humans. Being the middle child, being told that my mum and dad love us all equally.
So you fit down here somewhere?
Yeah, down on the left-hand side of the where Luke Stephen.
Oh.
Any questions for Sam? Guys, so we'll just go to Troy straight up.
Sam, I'm not sure if this question's for you or for someone else, but let's just say I'm testing the 20% of my males, like we've sort of been suggested.
Yep.
if I'm testing those animals, will I get information on, say, eating quality traits on related animals in the same drop? So through pedigree, can you predict?
So at the moment, no, you wouldn't. But when the single step analysis comes along, you would. So that's the important thing about single step, is that it starts to propagate through all of those animals that you've measured through pedigree as well. So you would start to get some information for those other sons as well if you had half sibs.
Any other questions? So what we might do now, guys, is that we still just have a bit of time before afternoon tea. So I might bring Sam and Steve back out here again and just sort of run for another 10 until smoke just on in terms of some of those key genomics talks. I know a lot of the things that we've seen today are around this application side of things. Has there been sort of any light bulb moments for someone today that they'd be keen to sort of share with the group based on what they were currently doing and potential of genomics? I think Andrew Heinrich's looking like he's...
Adrian, what was the question?
So applications of genomics based on what you've seen today. Of course, having a merino and a water something flock in terms of applications for your business.
Yes, I was thinking actually, I'm thinking the last, our 2013 drop, we actually did 20% of our merinos with genomics and used 3 round lambs, so that was pretty cool. Pretty excited about that. We've done a little bit of our whites, and I'm a bit like most of us terminal breeders, we'd like to get that information back earlier. And sitting there, I was thinking, I'm going to have to do 20% of my whites as well, and I'm going to have to fork out $3 grand to do that. So that's what my take home is of today, and hopefully we can get some of those curve benders, if we do that 20% on eating qualities with the build them, that'd be really cool. Just back to Deb.
Can I just ask Sam when the single step is likely to happen? Because I was just wondering if that's the case, and I took it to mean that perhaps in that situation we might not need to test siblings, like how closely related would we not need to test an animal in order to still get information back? So like we might be able to selectively choose the 20% that we test, and in effect get a whole lot more information back because they're related. Do you know what I mean?
Yeah, I can see what you mean, but you won't, even though there'll be some information propagated to the other half sibs, you won't get 100% of the value you would have got by testing each one of them. Does that make sense? But it's not a bad idea to see that there is information flowing both ways, but you'll still get more accuracy if you test them all. That's just how it is. As for your question with how long until the single step actually comes up, that's probably a Dan Brown question, because I don't really work for AgBoo, and depends on how long it takes for them to organise the models within OVIS and sheep genetics.
Yeah, thanks Sam. We are actively working on that, and literally we're starting very shortly to do some test analyses and start to understand and validate single step analyses. So it is actively working, watch this space, but it will take a little bit of time to get everything working in terms of the routine ASPV runs. But we do hope to have prototype runs out within the next 12 months to get a feel for what they'll look like and what sort of issues we might deal with in the routine evaluations.
I'll follow that question up with another question for Sam. Sam, if you've got 2 outstanding full SIP brothers, would you test one or would you test both of them, and why?
So I would test both of them, because at the moment you could you could, even if it was through pedigree, you'd think, if I test one, he gets only this amount of information for this reason exactly. That full sibs don't, that Luke is a bit less related to his full sibs than he thought he was. And so he actually sits down this end. He's really different to his brother, whose brothers who are quite similar. And they're up this end. They 60% of their genes are the same, whereas poor old Lukey up here, he's only got 40% of his genes the same as his brothers.
And that's why one of my brothers is a news presenter, and I've got a good head for radio. So.
I think you've got a bit of a sob story here. It's all right.
I just felt Tom was getting a lot of laughs.
Yeah, so in answer to that question, I'd test both, so you could actually differentiate between those two full sibs. Because with pedigree, they get without any phenotypic information, they get exactly the same breeding value. And if you test both of them, you can start to tease them apart, as Steve said. Yeah, I know. It'd be actually interesting to see how much information is actually propagated from genomics to pedigree, because that's still a bit of an unknown quantity of exactly what would go where. And so it'd be interesting to see, or maybe our modelling friend Steve Lee could have a bit of a look at a way of using that information within families.
Sam, Lee Hardwick from Wangaratta. You probably can't answer this question, but like everyone's looking for that curve bender with great meat-eating qualities, great ASBVs. But from a commercial point of view, if you test, or however many rams you test, if your best meat-eating quality ram comes up with a, let's say, a ASBV of post-weaning weight of plus 15, and your top ram's plus 18, commercially, which is that too, like, where's the cut-off point where you're losing money by picking from meat-eating qualities rather than ASBVs? And hopefully there's studies looking at that, because there's got to be a measurement of what's too low, and like, are you gaining in one area but you're losing drastically in other areas?
Yeah, that's a really good question, and it relates to how you weight traits within an index when you're making selection decisions. So let's not even talk about meat eating quality, if it was just weight and fat, and if you selected on weight and then came through and selected on fat later, it's the same theory behind. But what you need to do is also take account of the correlations between the traits and the best way and economic values of each trait. So if post-weaning weight is worth a lot of money and eating quality is not worth a whole lot, then it may be better to select for more post-weaning weight. But I don't know the exact economic values or genetic correlations between those two traits to really comment on which you should place more weight on than the other. And I think that's an important thing for sheep genetics to kind of get a handle on, is what are those relationships between those traits and how does it relate within an economic value. Sam might have something else to say too.
So Lee, as part of the sheep CRC meat program, they went and did some survey work on MSA standards, or the MSA star ratings, and you would have seen those slides over the last couple of years in terms of price differentials for the different star ratings. What else has happened is over that time there's been around 4,500 consumer eating quality scores taken on the INF progeny, the information nucleus lock progeny. That's now at a point where the meat scientists and agbo are starting to look at that in terms of using it as a breeding value, look at the correlations to the lab measurements, which is IMF, shear force, meat yield, and looking at the correlations to the production traits, so growth, fat, muscle. So Daniel and his colleagues have already done the lab work against the productive traits, so IMF to fat, shear force to muscle, and look at the correlations there, which are quite interesting. The next step is taking to the next level, which is the consumer eating quality scores, putting all of those together into the single step analysis, so all of that information feeds into each other, and then combining that in the selection index.
done some models there with the, we did that one with the white suffix there, and it was the rate of gain over the last 10 or 11 years, or what it was. But they've done models when that rate of gain will actually cease to be the big leaps we're making.
Was that the graphs that Richard put up before?
Yeah, I think it was.
So what happened there, and when Richard put up the changes in genetic trends over time for growth, fat and muscle, and there was a red line that came up and showed a differential rate of change. That was when the new Carcass Plus / LAM 2020 index was introduced. So prior to that, Carcass Plus was, a desired gains balance index, which had 60% on growth, 20% on fat, 20% on muscle. Over time, that started to put more and more pressure on fat, which was really, really good for a lean meat yield trend, which Richard put up, but less useful for managing eating quality. So around 5 or 6 years ago, the indexes were changed so that there was more selection pressure put on muscle and less on fat. So that slowed the rate of improvement for lean meat yield, but it's basically meant for the last 5 years, eating quality has been maintained within the sheep industry, just through those traits. The advantage now is that you can now directly select on those traits to improve them rather than wait on a relationship on your measured live animals that goes through. So the rate of change was a change of laws.
Evening was Wolf. Seeing that the Texels are such an island on their own, with supposedly so few linkages, where does that leave them in the position as far as genomics is concerned? And if introductions were made to be able to establish these, is it possible that being separated so much that the level of heterosis would be greater? And if it was, how would that be taken into account?
So there's a couple of questions within the question. The first part I take as, because they're a separate owned population, how do you deal with that when it comes to genomic prediction? There are two ways. The first way is what Sam talked about, is having enough animals measured to actually get a genomic prediction. The second way, which is only a possible way, it's not a set in concrete way, would be to try and test influential size and use some of those relationships to give you some extra benefit than what you would have got with pedigree. When it comes to trying to integrate them into the other populations, which I take as the third part of the question, that's quite an interesting concept. It's still going to be difficult because it's a smaller, more compact breed, and so you can't use information from the Pole Dorsets and the white Suffolks, even if you crossed them with them. You wouldn't be able to. You'd only be able to use that Texel portion of those animals that were bred. So if they were 50% Texel, and you had a Texel ram, you bred it to a white Suffolk ewe, you'd only be able to use the Texel portion of those genes to make that prediction of breeding value. When it comes to heterosis within that crossbred animal, you'd have to try and make estimates of how much the purebreds would have performed versus what the heterosis would be, and take account of that. But that's the only way you can do it. There's no silver bullet way of getting some Texel rams, taking them out to many, many ewes, and getting a big enough population to use a genomic prediction for.
I'm not sure who this question's for.
Where's it
I hope
come
I'm
from?
understanding correctly that the genomic information is particularly important and helpful where you can't get progeny testing results until later on, whereas in terminals you tend to get the progeny information earlier. Could somebody comment on how that plays into your maternal sires, and in a practical sense, should we be selecting our maternal sires at a different time from our terminal sires?
So, okay, it doesn't matter. Do you want me to, or do you want to?
I'll let you go first. So with maternals, the key benefit where genomics the place where there'll be the biggest benefit from genomics is in those lifetime reproductive rate traits. Once we have genomic breeding values incorporated into the ASBBs and maternals on them, I'd select at the same time you've been selecting and just have that component there. I've done some modelling around what's the benefit of that, and it's comparable to what I've been seeing in merinos. If the test was $50 and you actually had the reproductive rate incorporated into it, then you're getting around about 10 to 20 times the investment back as an industry over 20 years. Has that partly answered the question?
I might add a little bit to it. So traditionally, if you had those maternal ewes that you selected early, you may not have been getting as much information as what you could have been if you selected them a bit later. And so it becomes a bit like a dairy example. Because it's a sex-limited trait, only your ewes have the lambs that inform the rams. And so if you had to wait for a good, accurate breeding value for maternal traits, you would have to wait 5, 6 years before those daughters of those sons have good records as well, and plenty of daughters. If you can start to build a reference population that incorporates that information, then you can select just at the normal young time, same as terminals, if you have a nice, accurate breeding value. But if you didn't, and you wanted an accurate breeding value, you'd have to wait a lot longer. Does that end? Yeah.
Yeah, I think you're saying that your maternal size selection can pretty much be at the same time as your terminal size selection.
Yeah.
Is that right?
If you have if you have a good, accurate breeding value, yes. Yeah.
So just following on from that, in sticking with the theme of breeds mattering, one of the things that we're seeing moving forward, I guess, is composite breeds, and in particular now white Suffix are starting to be used as a sort of maternal base. Does these sort of the graphs you showed today and some of the things you're suggesting, I guess really open the market there for those sort of things?
Yeah, so when it comes to composite breeding, if you have the genes of the composite breed within the reference population, then you can still predict those composites quite well. And if we take our Pole Dorsets and White Suffix, for example, there's quite a spread within the breeds already that they actually have genes from other breeds located within them for the last few years anyway. And so if you have records of progeny of some of those rams or some of those lines, then you can use that information to predict composites, to predict terminal composites, maternal composites, along those lines. If you wanted to do what Luke said and take some rams from the terminal side and put it into the maternal reference population and get an accurate prediction, you'd have to make sure that you had some of those animals that came from that terminal side within the maternal run, because otherwise that information wouldn't go across because you use the reference population separately.
We'll just go, Barry.
Yeah, this one's for Steve. Have you done any of the economic modelling on the benefits of the genomic testing in the terminals?
Thanks, Barry. And yes, I have. The benefit's not as great using the current indexes. So I think that the key outcome that I've been primarily working with merino producers to now. Once we actually start getting an idea of what those genetic correlations are between the eating quality and yield traits, and incorporating that into the breeding objective, and understanding how much of the genomic, how good genomics is at predicting those traits, I'll be knocking on terminal breeders' doors very soon. So the answer is it's coming. Okay, just go to Doug, and then one last question, and we'll break the session.
Yeah, Doug Picker, just to you, Sam. Say this particularly for merinos, if you've got a stud ram that's got reasonably good ASBB figures on himself, but nothing yet on his progeny, is there any value in getting a genomic test on him, or are you just better off waiting the next year till his first drop of lambs there and just do them? Or if you had him done, is there any benefit in improving his lambs figures?
So if the size already got accurate breeding values, as you saw from the little breakdown of how the genomic test contributes to the breeding value, it doesn't make too much of a difference in terms of accuracy. better off waiting for the progeny to come through and, you know, following it through that way.
Look, if there's no more questions, I might just draw this session to a close. So thanks, Sam, Steve and Sam, for working